All right, so I split this video into two. Hopefully you took a little break. There's a lot going on here in chapter 8. So we're going to jump back in. And what I want to do is kind of talk about a few kind of, I guess, unique things that bacteria do genetically, and then we'll go ahead and dive into biotechnology, which is a big portion of the second half of your chapter. Okay, so in bacteria, we've got what's called horizontal gene transfer. Now, what we're more used to as humans is vertical gene transfer, right? You make babies, they inherit some of your genes, right? And so how are things passed on generation to generation? Horizontal gene transfer then means that you can give genes to your brother or sister, right? Or a cousin. This is weird. This doesn't happen in humans. I shouldn't say it's weird. Bacteria do it, and bacteria are very normal. So, but a lot of times we don't get to talk about bacteria in Gen Bio as much. So there's three main types of horizontal gene transfer that we're going to look at: conjugation, transformation, and transduction. So let's take a peek. So here's conjugation, right? So these bacteria are going to use that conjugation pili, or pilus, singular, or sex pilus, right? And they then are able to make this bridge between their neighbor, right, a cousin or something, and hand DNA in there. Now, typically, what they're going to hand off through that pilus is a plasmid. So here we see actually making a quick copy of that plasmid and donating it to a neighbor. Okay. Same thing showing up in this picture, except now what we're doing is giving them a portion of the chromosome. Now, the thing to keep in mind with this conjugation is that often what bacteria are passing on to their neighbors is actually really useful. So, excuse me, antibiotic resistance and things like that. Transformation. So this is another form of horizontal gene transfer. And what we're going to see here is that bacteria have the ability to pick up naked DNA in their environment. So if they're hanging out in your sink and other bacteria have died and left some DNA behind, they actually have receptors on the surface of their cell where they can pull in samples of that DNA. And if it's useful, they might just go ahead and hold on to it. We don't do that. Okay, but it can be really useful because they might pick up DNA that gives them a trait that's advantageous. This is kind of a classic example of transformation, and actually the study that helped scientists and people realize that DNA was actually genetic material. So basically what you have here, I feel like I've talked about this study before. Maybe that was in Gen Bio. So here we have a Staphylococcus species. It has a capsule. Remember that capsule often makes bacteria much more pathogenic. And so if we infect this mouse, it dies. There's another version that they're calling the R strain. What do they call this one? The S strain with the capsule. The R strain doesn't have a capsule, and when you inject it in the mouse, he's totally fine. Okay. If you heat kill the S strain, the one that should be pathogenic, right, the one that has the capsule, the mouse survives, right? It's dead bacteria. So that makes perfect sense. Shouldn't kill them. Here's where it gets weird. If you put the live, non-pathogenic strain, right, one that shouldn't harm a mouse, and you mix it in a test tube with the heat-killed strain, which shouldn't kill a mouse, and then you inject it, you kill the mouse. Why? Right? Notice what happens. If you look at that dead mouse, right, and you isolate the bacteria that were in there, there are some capsule-forming bacteria. And so this is what transformation is what explains that, right? These live R strains were able to pick up some of the DNA and gain the instructions on how to make a capsule, which allows them to be more pathogenic. And so that's some of why we're interested in things like conjugation, transformation, transduction, is it can change the pathogenicity of our microbes. Okay, so then the third and final form is transduction. And this is actually when a phage, remember bacteriophages are viral viruses, are bacterial viruses. They are viruses that only infect bacteria. So here we see the phage inserting his DNA, and we're expecting then the bacterial cell machinery is going to take over and produce new copies of that phage. And I know this is kind of small, it's difficult to see, but notice the phage DNA was like dark red and the bacterial chromosome is this lighter pink. Notice what happens when these phages come out here. This one's actually carrying some bacterial DNA. So in transduction, we're looking at the ability of a phage to move DNA from one bacteria to another. So that's also horizontal gene transfer. Okay, continuing with this thought on genetics, this chapter gets into how mutations take place, and I wanted to point out that some mutations are inherited and others are actually spontaneous. They're just like a mistake in some form of copying, like a DNA replication process. But then some mutations are caused, right? So we have what are called mutagenic agents, things that cause mutations. They can be chemical, biological. Here they're showing physical things like radiation, right? So the reason I try to point this out is to realize that your risk of mutation in you, and therefore your risk of something like cancer, is somewhat associated with genetic inheritance, but absolutely associated with your environment. Okay, and so we've surrounded ourselves with lots of different chemicals that can mutate our DNA, and it might be in our best interest to avoid them. The easiest one to avoid would be the hundreds of chemicals that are in cigarettes. Okay, so when these mutations take place, truly, technically, a mutation is just a change in the DNA. And we can have very minor changes, and we can have more drastic changes. So look at this. What they're showing you here is the DNA code, and then the RNA code, and then the amino acid, right, that codes for. So here's our normal gene, right, our normal DNA, how the RNA and then the protein is built. We've got a couple different options here. In a missense mutation, it can be, so a point mutation is if you just have a single letter that changes. If a point mutation makes, if any kind of mutation makes it so that the amino acid in the final protein differs, that's considered a missense mutation. So if it causes the change of the amino acid, it's missense. You can also have a silent mutation, right? We saw the redundancy in that table. You could have a point mutation and still get the exact same amino acid. That would be a silent mutation. And that's what they're showing here, right? Is this, we got a change, but we still ended up with, what is that? Aspartame? ASP for the amino acid, right? So it was a silent mutation. All right. Frame shift mutations tend to be a little worse. So this can either be from a deletion or an addition, but notice what happens here. So it was supposed to be TGGCTG. We remove a G, and now how we read the codon shifts over. We're still going to read 3 letters at a time, and we don't realize that we've lost this G. And notice what happens now in this frame shift mutation. Boom. We start just like every single amino acid is wrong. Okay. This can also then lead to the premature, so that protein's not going to work. And then sometimes it also leads to a premature stop codon. And so often this amino acid is way shorter, or sorry, this protein is way shorter, and it's not going to function. That can also happen from an insertion, right? Notice how we added an extra G here. And again, that changes the amino acids being put into the polypeptide. This is something called the Ames Test. This is a test of the mutagenicity of a substance. And so before you even get to like studying, a new makeup on people or rabbits, you would first want to do this Ames test. And so basically the way this works, this is a little confusing, but check out what happens here. So we are going to grow salmonella. Okay, and we grow a specific strain of salmonella that is histidine negative. This means that it cannot produce histidine. In order to survive, we would actually have to put histidine in the agar, right, for it to live. So we've got a control plate and a test plate, and basically They're both what's called minimal media, so there's not a lot of nutrients in there. And specifically, notice that both plates are lacking histidine. But on the test plate, we're now going to mix in our new, foundation, some chemical that's going to make the best, makeup ever. Okay, so notice what happens. We're incubating this. Nothing should live, right? This bacteria can't make histidine. It needs histidine to survive and reproduce, and we haven't given it any. The only way bacteria can survive is if it has a mutation, and it's a back mutation, if it has a mutation so that it can produce histidine. And this isn't that uncommon because bacteria reproduce so rapidly, these kind of mutations show up. So sure enough, on our control plate, we get a couple colonies. Those bacteria had some spontaneous mutations, right? And they were able to survive. Whoa, but notice what happened over here. We have lots of colonies. That means lots of our bacteria mutated in response to this chemical, and now we're able to survive. Okay, and so again, this is the Ames test, A-M-E-S. So kind of handy before you get too far along in releasing a product, trying to decide what its mutagenic potential is. Okay, so the second half of this chapter is really all about genetic engineering. And I did want to remind you guys, you have a paper coming up here. The second paper deals with genetic engineering with biotechnology. And so what we're getting into with genetic engineering is the direct, deliberate modification of an organism's genome. Okay, this is typically a really important part of biotechnology, but other things can be considered biotech as well. But biotechnology is when you manipulate an organism or one of its components to produce a useful product. So some of our most amazing biotechnologies absolutely have come through genetic engineering, but not all genetic engineering is biotechnology, and not all biotech has to deal with genetic engineering. Okay, so in genetic engineering, as you're looking at this part of the chapter, look for things that are interesting to you, things that you might want to write about a little bit more in depth. So we'll run through some of this really quickly. A lot of this goes through kind of different lab techniques, different technologies we have as part of kind of our DNA technology toolkit. Again, hopefully you used some of these in Gen Bio. Hopefully you did some gel electrophoresis and used restriction endonucleases, but let's run through here real quick. So this is trying to show us restriction endonucleases. Oh, they started out with denaturing. Okay, so one thing that we use in DNA technology is our ability to denature DNA, to get it to fall apart, right? We take a double helix, we can make it split into two simply through heating. As soon as you cool those two strands, they're going to find each other and go back together. Okay, so we use denaturation. We also use a huge set of enzymes, particularly these ones that are called restriction endonucleases. I like this little picture here. That's exactly what they do. They act like a little pair of scissors. And the restriction endonuclease is going to cut the DNA at a very specific spot. So there's actually hundreds, maybe even thousands now, of different restriction endonucleases that we know cut in slightly different areas. They point out that the way these work, there's always a palindrome, right? It's the same in both directions. They're pointing out these sticky ends. And when I say it's the same in both directions, notice GATC, and then on this side, GATC. So there's what's called sticky ends. If you cut, say, human DNA with a restriction endonuclease, and then you cut a bacterial plasmid with that same restriction endonuclease, you would get complementary sticky ends because they always cut at the same sequence. So this is a cool technology you can use to actually make these recombinant DNA situations, right, where you can move DNA from one organism to another. We also use this in Electrophoresis, again, if you cut up DNA, this is something you could use on like a paternity test or a crime scene. If you cut all your samples with the same set of restriction endonucleases, it's going to cut it into these different size chunks depending on the DNA code available, and then you can look for where you find matches, right? What was at the crime scene, which suspect actually matches that. So we can use that to analyze DNA. That's a really big analysis. We also, this is a technique called PCR. I think I've mentioned this before, where you take a DNA sample and maybe, again, at your crime scene, it's a really small sample and you want to amplify it. You want to get many copies. And so basically you can take that DNA sample, get it to denature, add all the nucleotides, the primers, the enzymes you need, and it will copy itself. And you tell it to copy itself over and over and over again, and sure enough, you can go to 1 fragment to 64 fragments, right, and on like that. In particular, I think I mentioned we sped this up with the realization that we could use Taq polymerase. We could use an enzyme from an aquatic Thermus aquaticus, from a hot spring organism, so that we could just heat up the DNA once and then the enzyme worked under those high temperatures. So again, this was kind of, this dramatically changed DNA technology. So again, PCR, polymerase chain reaction. You might see that too. You're testing someone, say, for a virus. This is really common. You don't have a huge sample. You need to amplify that sample before you can run tests. Okay, so then we get into gene cloning. So what this is looking at, again, is a recombinant organism. So we have a genetic donor here. We're going to find the gene we're interested in and we insert it here into a plasmid, right? We can then give that recombinant plasmid, some of it's bacterial, some of it's, say, human, to bacterium. And then that bacterium will go ahead and undergo transcription translation and make the protein. And this is where I was saying you do have to make sure, and this kind of shortcutted it, if you want a bacteria to make a human protein, you can't just give it human DNA. You need to get that mRNA go back to DNA, and then you can make sure that the introns are being removed and then an actual functional protein is being produced. But this is huge in biotech and genetic engineering. We now have bacteria, I think they mostly use E. coli, in huge vats that can produce human insulin, human growth hormone, certain clotting factors that maybe a hemophiliac needs. Really crazy that we can use bacteria as little factories to produce the proteins that we're interested in. Okay. I think, so what I was going to say, so the rest of your chapter then, there is more, right? They talk about genetically modified organisms. Typically, we're thinking about things like crops. I mean, that bacteria we just saw making insulin is genetically modified, but thinking about things like crops. Gene therapy, I imagine a lot of you would be interested in that, right? Turning these genetic modifications into therapies for patients. And so look around, look through the rest of the chapter for ideas. We'll see what people write about. We'll talk about those in class, and then I'll try to fill in any gaps that people don't really cover on some of the important biotechnologies. If you're having trouble, feel free to pick in a topic. Feel free to shoot me an email. I'm happy to chat or give you some more ideas on something you might be interested in. Okay,